So transduction transformations that are already explained by me in my channel, and so you just check out that all. And this is the process, that is conjugation. Okay. So here I want to say thank you to all of you who have already subscribed my channel and support me a lot for the growth of this channel. Okay. And I want to say to the newcomers, if this channel is helpful to you, then please do subscribe and share this channel with with your friends. Okay. So conjugation process is what? This conjugation process is that the direct contact between the donor and the recipient. Okay, donor and recipient. Recipient bacteria and leads to the establishment of cytoplasmic bridge between them and transfer of a part or all of donor genome to the recipients takes place. Okay. So, one second. Sorry, I just checking the thing that was missing here. Okay. So, what is saying that in conjugation process, this is a direct contact between the donor and the recipient bacteria, and that leads to the establishment of the cytoplasmic bridge between them and transfer of part or all of donor genome to the recipient takes place. So, here actually suppose this is the if these are the two bacteria, then here the transfer of the DNA is possible by making a bridge. Okay, by making a bridge. So this is suppose one bacteria and this is suppose another bacteria. So DNA that is transferred in this way in the recipient. Okay, so I just explained this diagram after explaining this. Okay, this is a very brief explanation that the conjugation process, actually here the transfer of DNA that is possible only by making the bridge between the two bacterial cell. One is the donor cell, another is the recipient. Donor cell that transfer the DNA and recipient that receive the DNA. And here the donor ability is determined. How the donor ability is determined? By the presence of self-transmissible conjugative plasmids, and this called sex plasmids. So the bacterial cells which have the conjugative plasmids, or you can say that the self-transmissible plasmids, then these are known as the donor. That means they can easily transfer and their DNA into the recipient cell, and the recipient cell that has received the DNA as a result of conjugation is called the transconjugate. So recipient, another name of the recipient cell is that the transconjugate, because by the process of conjugation they receive the donor DNA from the donor cell. Many plasmids, that means here the conjugation process that is very intraspecific. That means intraspecific, that means the same bacterial species or the conjugation process occurring in the same bacterial group. So here sometimes many plasmids have the transfer system and that enable them to transfer between unrelated species, and these are known as the promiscuous plasmids. Promiscuous plasmids. So what is saying that generally conjugation process, that is the intraspecific process. Intraspecific means that is the such type of group or individuals that in the bacteria there the conjugation process is occurring within one bacteria to another bacteria. But in many cases, the plasmids have the transfer system by which they can transfer DNA in the unrelated species, and in that situation, these are known as the promiscuous plasmids. Okay. So how the conjugation process occur in the F plus and F minus conjugation? So generally three types of conjugation is seen. One, the transfer of DNA from F plus to F minus conjugation. Another is the transfer of DNA from HFR to F minus conjugation. And the last type of conjugation where the DNA transfer from the F dash and F minus bacterial cell. Okay. So here, first is that the F plus and F minus conjugation. So before going to the expansion of the conjugation, first we have to understand what are the F plus and F minus plasmids or the cell or F plus mean what or F minus mean what. So that F plasmid, it is the F factor, and this is prototype for fertility plasmids in gram-negative bacteria. So F means this is the F factor, that is prototype for fertility plasmids. And if the bacterial cell that containing this F factor, these are called the F plus, and which haven't F factor, these are known as the F minus. So what is said that strains of E. coli with an extra chromosomal F plasmid, these are called the F plus, that means the F factor present here, and these functions as donors. Okay, and the strains that lack the F plasmid, then these acts like the F minus and behave as the recipients. So you just clearly understand this part. I'll just briefly explain again. Conjugation process is that where the transmission of DNA is possible by making a bridge between the two bacterial cells, and obviously in case of F plus and F minus conjugation, there is the F plus that is considered as donor and F minus that is considered as recipient, and F plus that has the F factors and F minus that haven't any F factors. Okay. Next, the conjugative functions of the F plasmid are specified by a cluster of at least 25 transfer or the tRNA genes. So F plasmid, they contains about 25 transfer genes, and which determine expression of sex pili, synthesis and transfer of DNA during mating. Mating interference with the ability of F plus bacteria to serve as the recipients and other functions. So generally the F plasmid that containing some group of genes, these genes are called the tragenes, and what is the factor of or a function of these tragenes? This determine the expression of the pili or the sex pili and the synthesis and also transfer of DNA. Three types of functions that is occurred by these tragenes. One is the expression of sex pili, synthesis and transfer of the DNA. Now, the tragenes that can be divided into two groups. Those whose products are involved in the mating pair formation, or you can say the short form MPF, and those whose products are involved in processing the plasmid DNA for transfer. So these are called the DTR. So one is MPF, another is the DTR. That is the two groups of tragenes involved in the two functions. One group of the genes that or the products of one group of genes that actually involved in the mating pair formation, or it helps in the formation of the mating pair. Okay, and the second products or the products of the second group of the tragenes that are involved in processing the plasmid DNA. Clear? So MPF, that component includes a 6 pillars that extrudes from the cell and holds the mating cells together. So what is the function of these pillars? This generally pillars is the singular form and you know that pill is the plural form. So what is the function of the pillars? It just help togetherly or attach together the two cell with each other. Okay, its function is that it tries to togetherly act or together. Actually its main function is that by this formation or by these pillars, the two cell holds each other. Okay, the two cells hold each other. And the MPF system also includes the channel in the membrane through the DNA and protein pass. So what is saying that it also helps in the formation of one channel through which DNA and protein may be passed from donor to recipient. Okay, now we will learn about how this F plus and F minus conjugation process is occurred. Okay, so continue the second part. Okay, so here the conjugation process of F plus and F minus is briefly described here. Now you see that here the transmission of DNA from F plus to F minus, and it is the very rough diagram drawn how the transfer of the plasmid from F plus to F minus is occurring. Okay, so each F plus bacteria that has generally 1 to 3 sex pili and that binds to specific outer membrane protein or recipient bacteria to initiate the mating. First of all, the two bacterial outermost membrane that containing protein, these are attached with the help of this 1 to 3 sex pili. Okay, and in that way the donor and the recipient both hold each other, or you can say that stabilize each other. And intracellular cytoplasmic bridge, you can see here the rough diagram. Here the cytoplasmic bridge, this is the cytoplasmic bridge. Okay, so cytoplasmic bridge is formed and one strand of the F plasmid DNA is transformed from donor to recipient. Okay, this is the F plasmid. Okay, F plasmid, and this F plus that is containing F plasmid and F minus that is not containing the F plasmid. In the first situation, here the nicking is seen in the one strand of this F plasmid, and after that the beginning of the transfer, or the you can say that the transmission of the DNA that becomes start. Okay, the one strand of the DNA only transfer from the donor to the recipient, and obviously here 5 prime to 3 prime direction that is maintained. Okay, and the site of the plasmid DNA at which the transfer initiate, this is called the origin of the transfer. So the site on the plasmid DNA where the transfer initiate, okay, the transfer where the initiate, this position is called the origin of transfer. And here one important protein, that is the relaxase, that makes a single strand cut at the origin of transfer region, and the transferred strand is converted to circular double-stranded F plasmid DNA in the recipient bacterium by the process of, you can say that, replication. Both of the X conjugant bacteria are F plus and the F plasmid can therefore spread by the infection among the genetically compatible population of the bacteria. So, what is happening here? First, one cut is seen in the one strand or the single strand of F plasmid. Okay, and then the transference of the single strand, it is seen, and that is occurring from donor to recipient. And what is the function of the relaxase protein that helps in the single strand cutting process? Okay, and after transferring of single strand from the donor to the recipient, here the replication process becomes start and it becomes double stranded here. So in the first condition, the first one is the F plus and the second one is the F minus, but after the complete process, the two bacteria that converts into, that not converts, the F minus that converts into the F plus bacteria. Okay, that means it containing the F plasmid. So this is the conjugation process, and by which the F plasmid transfers from the donor to the recipient. Okay. So another two processes obviously related to the conjugation we have to learn, but here I stop this video with the first process, and I will try to explain the second two processes in the second video. Okay, so you just continue with my channel. Okay, thank you.